Good morning, ladies and gentlemen. My name is Anna Rygula and I'm from National Museum in Kraków. I would like to thank for the invitation and the opportunity to present a result of our project. I would like to talk about non-invasive analysis of the pigment palette used by the Renaissance painter Sofonisba Anguissola. Sofonisba Anguissola was born in 1532 in Cremona as the oldest daughter of Bianca Ponzoni and Amilcare Anguissola. Her first teacher was Bernardino Campi. Initially, she mostly painted small religious paintings, but quickly she became noticed as a portrait painter. Sofonisba was 30 years old when she became famous as official court painter to the Spanish king Philip II. We focused the project on the analysis of the painting Self-Portrait at an Easel, which is in Museum Castle in Łańcut in Poland. The palette used for Self-Portrait at an Easel was compared to another famous painting from the same period, The Game of Chess, which is in National Museum in Poznań in Poland. In the painting Self-Portrait at an Easel, Sofonisba Anguissola shows herself in three-quarter view while working. The artist looks at the viewer. She is dressed in dark clothes with white lace collar and cuffs. The background is dark green, and at the left there is an oil painting on an easel. She's painting Madonna and Child. The project was carried out applying both macro and micro analytical techniques. Because we couldn't take samples, we had to focus on the applying a non-invasive techniques. Firstly, we apply a technical photography using UV radiation and X-radiography together with macro XRF to better understand the materials and techniques. Then, based on the macro results, we choose the measurement points for the microscopic non-invasive techniques like single-point micro XRF, micro-Raman spectroscopy, and optical coherence tomography called OCT. All techniques were used as complementary methods to enhance the level of detail of results obtained with macro techniques. X-radiography were obtained using DI X-ray system, which is based on a wireless panel. And UV luminescence photography were done with Canon EOS with lens 100 millimeters, with fluorescence tube with a maximum at 368 nanometers. These dark areas, clearly visible here, means quenching the fluorescence, what is connected with a retouching of these areas. Two-dimensional macro XRF scans of the painting were carried out using an M6 jet stream spectrometer from Bruker, and the pixel size of the elemental maps was set to 2 millimeters with the beam spot size of 650 microns. Macro XRF was performed for visualizing the distribution of elements which are contained in the pigments used throughout the painting. Micro XRF were conducted using an Artax 800 spectrometer from Bruker with an analysis spot below 100 microns. As I mentioned, macro XRF was done for visualizing the distribution of elements, but to improve the detection limit for lighter elements such as phosphorus or silicon that could not be detected by the macro XRF, we use also the micro XRF spectrometer. Micro-Raman analyses were carried out using a Renishaw INVIA spectrometer. We applied the red laser and measurements were carried out using a steerable arm because the dimensions of the object make it impossible to measure it in the microscopic observation chamber. The steerable arm, here on the photo, enables to measure the object located outside the measuring chamber by an optical system placed as an objective in the microscope turret. The collection of spectra was difficult for pigment giving a weaker Raman signal because of the varnish layer covered the painting. The next technique is OCT, optical coherence tomography. This non-invasive technique uses near-infrared light of low intensity to show the internal structure of objects by localization of all the discontinuities and rapid changes of the refractive index of the tested medium. The results are usually presented as cross-sectional views. The tests were carried out using a prototype portable OCT instrument constructed in the Institute of Physics in Nicolaus Copernicus University in Torun. Now I like to present our results according to pigments and colors. We pay attention to the characterization of Sofonisba's dark dress. The single-point XRF was conducted to expand results gained from the macro XRF mapping of this area. During micro XRF analysis, lighter elements like phosphorus and potassium were detected, which can potentially provide a clear distinction between black pigments. The high concentration of potassium in dark areas provides evidence for the presence of a plant-based black pigments. Also, the presence of calcium visible on the macro XRF maps, together with phosphorus observed by micro XRF, suggests the presence of bone black. The application of both black pigments together seems to be characteristic for the work of Sofonisba Anguissola. Macro XRF images of calcium and potassium suggest that Sofonisba preferred a vegetable-based black pigment for brownish colors, for example, sleeve of the right hand, whilst for more black tones, the bone black was preferred. The black pigments were mixed with Getit, lead pigment, probably massicot, and in traces with copper pigment to obtain the right shade of brown. The detection of lead in white areas of the painting layer serves to identify the nature of pigments used to paint these zones. The two pigments with lead were identified by Raman spectroscopy in skin tones, lead white and massicot together with vermilion. However, lead white can be applied both in the painting layer to paint brighter areas, exactly like massicot, and also in the ground layer to obtain a white and smooth surface to draw paint and to limit absorption of oil binder. The first type of application of lead white can be observed on the diffraction map showing the presence of lead in superficial layers, where this pigment is applied on the skin tones. But also in light yellow and blue areas, which was confirmed by Raman spectroscopy. The use of lead pigment allowed to observe some of the author's correction in an area of Madonna's painting in the X radiographs. The originally painted picture of Madonna with child was wider and lower. It's marked here with blue arrows. These changes were also detected by OCT. Moreover, this is the next evidence that lead white was added to the gypsum ground layer. X radiographs shows the structure of the canvas, which may provide an evidence about the presence of lead in the ground layer. Here is OCT image showing the author's correction. The central panel, this is location of the scan in visible light, and the right panel is the same over the X radiograph. And the OCT scan is here. Number 1, it's upper varnish layer. Number 2 is bottom varnish layer. 3, semi-transparent paint layer containing smalt. And 4, this is underlaying paint layer. And this is probably the primary composition. The calcium pigment is gently visible on whole canvas. This is evidence for the application of gypsum in the ground. The areas on the calcium map where parts of the complexation and painted picture are black are caused by the absorption of calcium signal in the overlaying lead-containing paint layer. Traces of retouching draw attention by the brighter signal of calcium. A high concentration of this element can be observed in two areas of past restoration treatments, under the painter's left shoulder and around the edges of the pictures. These areas are clearly visible in UV-induced fluorescence imaging as dark spots. It can be concluded that the retouching was made on a chalk or gypsum-based putty. Based on the macro XRF mapping and single-point micro XRF measurements, it was possible to conclude that the dark green background was obtained by using copper and iron pigments with bone and or blend black together with traces of vermilion. According to Raman spectroscopy measurements, the background color consists of a copper-green pigment or pigments mixture based probably on the chlorides, which corresponds to the variation of the popular green pigment verdigris. To obtain the dark brown hue getted with its natural impurities was applied in the painting layer. The characteristic bands of getted were observed, but also the accompanying minerals were detected like anatas and jarosite, which are minerals of volcanic origin that appear in the literature as admixtures in getted deposits. The weak band at 153 comes from the lead-white presence, while vermilion is characterized by bands near 252. These broad bands come from carbon black. The blue part of the Madonna's dress was painted partially with smalt pigment. This conclusion is supported by the presence of cobalt, nickel, and arsenic detected by XRF. Moreover, the Raman measurements revealed also the use of indigo pigment. Moreover, traces of copper were detectable by single-point micro XRF measurements, providing evidence of the application of copper blue or green pigments. The main red pigment used by the artist is vermilion, mercury sulfide. The distribution of mercury is presented as a result of the XRF mapping. There is surprisingly little of it in Madonna's dress, but its presence in this part was confirmed by Raman spectroscopy. The bright red part of Madonna's dress was painted probably with an addition of organic pigment, but is not detectable both by Raman spectroscopy and XRF techniques. The semi-transparent layer of organic pigment is observable in the OCT image. The painting Self-Portrait at an Easel by Sofonisba Anguissola was compared to the game of chess by the same artist. The core palette in the two investigated paintings consisted of vermilion, lead white, smalt with indigo, getted with impurities like jarosite and anatas, vegetable and bone carbon black, a copper green pigment, and massicot. The pigments identified in these two paintings are in agreement with those employed by Renaissance painters during this time period and at the geographical location. The results of this work demonstrate that the combination of imaging and spectroscopic techniques can be used as a non-invasive diagnostic methodology to investigate the upper layer structure of artworks. Finally, I would like to thank the co-authors from Laboratory of Analysis and Non-Destructive Investigation of Heritage Objects in National Museum in Kraków in Poland. And I would like to thank also co-authors Alicja Mogielska, Professor Magdalena Iwanicka, and Professor Piotr Targowski. Results were obtained within the MOLAB Fixed Lab project of the Polish Research Consortium for Heritage Science, HERI-TECH. Thank you very much for your attention, and I'll be happy if you have any question or suggestion.